So we're here at the Biologists at 100 conference with Giulia Paci, and you have a preprint out currently on arXiv, is that right?
Yes, exactly.
So I was wondering, could you begin by telling us a little bit about your preprint in about a minute, if you can?
Yeah, sure. So what we've just uploaded is like a protocol paper. So I would say it's the fruits of labor of several years of optimizing a quite challenging imaging problem in the lab. And so we've put together an end-to-end protocol that explains how one can go about imaging through a deep and very dense tissue with fluorescence microscopy, but then also use computational tools building on many open access existing tools to do single cell segmentation within these very dense tissues.
Okay, that sounds like quite a challenge. And so given that you've been working on this for some years, you said, I wonder, over that time period, what was the most challenging aspect in terms of getting this to the preprint stage?
Yeah, no, that's a great question. I think the most challenging bit is going from, let's say, some sort of pipeline that works for you to actually making sure that you can set up something that's very reproducible and that anyone can use, let's say, from different systems.
Yeah.
And so that has taken quite a bit of time, and a lot of credit goes to my co-author Pablo, who's really put a lot of work into testing with different softwares and making sure that when we shared it, as much of it is like ready to use for people outside of the lab and anyone can test it.
Yeah, it's always nice when other people can use it as well, especially when you're putting it up as a preprint.
Yeah.
So you've been presenting this work at the Biologists at 100 conference, and I was wondering, in general, like either here or in general with your preprint, have you had any good feedback on it that's sort of affected the way you do your research? Or
Yeah, absolutely. So the preprint is quite fresh. So this is almost like, I would say, the first time I've been kind of showing that to people, and the feedback is great. So we've had a few people come. Actually, one person had already tested our pipeline, and in general, lots of people come with like very similar problems to what we were also trying to address. And so it's been like very productive conversations, and also really great to hear that people have been able to try it and give us feedback on how it's working for their own problems. So that's really great. Yeah.
And you said that this preprint's fairly fresh, but how do you anticipate maybe it might benefit you personally in like your career and your aspirations for the future?
Yeah, so this is, I think, is a great aspect of preprints. I must say I've already benefited from preprints throughout my career. So for example, in my PhD work, my PhD paper was the first preprint the lab ever published because it was still quite new. But thanks to that, I could apply for postdoc position. I could apply for postdoctoral fellowships because many funders now consider preprints. So that's already been great for me so far. And now I'm kind of preparing for the next career stage, right? Getting ready for my own independent position. Being able to put my work out there is great because it allows people to see what I've done without a delay of, a year on average for things to go through the whole peer review process. And so I think that it has already been great, and it will further benefit me in the future as well.
It's very nice to hear that they have really benefited you. And I mean, in your answer, I guess you kind of touched on it already, but what do you think the importance of preprints are for the research community and publishing in general, especially moving forward?
Yeah, so I think they've already been very transformative, and like just a couple of things I've already touched upon. So one is, for example, the fact that we can share things much more quickly. So in our case, again, this is a protocol tool. So the idea is we want to get it to people as fast as possible. It wouldn't really make sense for this to stay sealed behind doors for an additional year, especially, in a field that moves fast. So again, our tools do leverage on, for example, machine learning in a kind of area where things evolve very quickly. And there's a risk that by the time things are out, then you already have to revisit them from scratch. So there's a really big advantage, I think, on sharing things very quickly. And then the second thing is definitely the advantage on all of the trainees, career progression. And again, in this particular preprint, many of the authors are actually master students or summer students that contributed across the years through testing many different aspects. And again, for them, I'm sure this small thing can be a great point to leverage on when they're applying for PhD position or for their next jobs. And so in the end, it benefits everyone, I think.
Yeah, I would agree. Well, thank you very much for taking the time out to come and speak to us about your preprint and preprints in general. We really appreciate it. We'll put the link to your arXiv preprint in the comment section of this video, and I hope you enjoy the rest of the Biologists at 100 conference.
Great. Thank you so much for the opportunity. It's been really nice talking to you.
All right.